Hello, my name is Henne Holstege. I work at the Department of Human Genetics and the Alzheimer Center at the Amsterdam University Medical Center in the Netherlands. And today I'd like to tell you about our endeavor to identify structural variations that associate with Alzheimer's disease. So first, some numbers. As you can see, by 2050, it is estimated that almost 14 million individuals in the United States alone will suffer from Alzheimer's disease. And this poses a huge burden on society. So we really need to identify a cure for this disease. However, that is not going to be easy because when you look at the process of Alzheimer's disease, you see that the brain shrinks and that holes appear in the brain. And there will never be a cure that will bring back the cells that were previously there. So the only cure for Alzheimer's disease is preventing this process from happening. But for that, we need to predict before the onset of the disease who runs a high risk of developing Alzheimer's disease. And for this, we have the genome in our hands, which we can use to predict who will get Alzheimer's disease later on in life. We know that from twin studies that 60 to 80% of Alzheimer's disease risk is heritable. And the associated genetic variants give insight in the etiology of the disease, but they can also be used to predict the risk before disease onset. The heritability, as far as we know now, is partly explained by the haplotypes identified using genome-wide association studies. But with GWAS, we identify genetic regions and that there's something in these regions that associate with Alzheimer's disease. But the regions themselves are not causal for disease. We also identify rare coding variants using short-read sequencing. And these can be causal for disease, but they do not explain the heritability for the disease. In fact, a large part of the heritability has not yet been explained. And part of the reason for that is that important parts of the DNA have never been investigated in a case control analysis. And for example, the structural variants have been completely neglected. And the reason for this is that these structural variations are very difficult to measure. They're very repetitive and they're long. So with the sequencing techniques that we have at hand currently, this was not possible. So this means that large insertions and deletions and inversions have not been identified yet in a systematic manner within a case control situation. So here's deletions and inversions, insertions and inversions, and tandem duplications or dispersed duplications, or actually copy number variations. They may all have an effect on Alzheimer's disease. So in fact, when we look at what is already known with regard to structural variation in neurodegenerative disease, we see that there are several diseases that are actually caused by structural variants, such as the Huntington's disease or ALS or FTD. Those are all neurodegenerative diseases that are actually caused by a structural variant. So when you look at Alzheimer's disease, we really don't want to neglect the effect of structural variants. Thus far, 86 loci have been known to date to associate with Alzheimer's disease. So this is the GWAS hits. Again, these are regions in the genome that associate with the disease, and that there's something in that region that is actually causative for the association, but not the region itself. I'm going to show you some specific ones, and this is the ApoE. This is the strongest risk allele for Alzheimer's disease, and for this we actually know what is actually the causative variant. Here we show the PCALM region and the ABCA7 region, and I'm showing you these because we're going to zoom into these regions. But again, for most of these regions, the driving variant in those regions is unknown for most loci. So when we zoom into the PCALM region here, this is the PCALM gene here, and here is the locus, and you see that there's a haplotype that associates with Alzheimer's disease. But in that region, several structural variations also map. And you can see here, there's a very large structural variant here that maps right at the beginning of the haplotype that associates with Alzheimer's disease. And it could be that some people have a copy number variation here that is the actual cause for the AD association, and that needs to be identified further. Here we look at the ABCA7 region. Here it's this is a less clear region, but you can also see that there are there's a haplotype that associates with Alzheimer's disease. And in the middle of the ABCA7 gene here, you can see also a large structural variation. Here's the structural variation. And this is in fact a variable nucleotide tandem repeat. So I'd like to go into the variable nucleotide tandem repeats here because it's important. Here you see a piece of DNA, and I'm plotting here the repeat sequences on the DNA. And these may actually expand and contract. So when these regions are short, it's not really a problem. But when they become too long, when they expand to very, very long repeat, this may actually cause a problem for the DNA. It may actually kind of tangle up, and that may have a problem for the transcription of the genes in its vicinity. So it has been very difficult to identify the length of these repeats because short read sequencing cannot span the repeat because they can be really, really long. So to identify the repeat sequence lengths, we need long read sequencing. And that is what we would like to do. So to go back to the ABCA7 repeat, here you can see the ABCA7 gene again. Here you see exome 18, and here you see exome 19, and here you see a 25 nucleotide subunit repeat sequence. And Arne de Roek et al. In 2018 has shown that when the number of repeats becomes so long, expand to such an extent that it exceeds 5,200 nucleotides, then it associates with a 4.5-fold increased risk for Alzheimer's disease. So basically 5,200 nucleotide is a critical length that you don't want to pass. So our aim is to identify other repeat sequences that will similarly increase the Alzheimer's disease risk. And we're going to do this by comparing Alzheimer's disease patients and centenarians, cognitively healthy centenarians who have escaped cognitive decline until at least 100 years. So we already know that the heritability of Alzheimer's disease is 60 to 80%. And based on sibling relative risk of this cohort, we were able to identify that an estimated 60% of the chance to become a cognitively healthy centenarian is heritable. So we want to generate long read sequencing data from these two opposite ends of the cognitive spectrum. We've done previous analysis on this comparison. We have generated polygenic risk score based on the AD GWAS loci. So what we can do is we can identify or calculate a score based on all those 86 GWAS loci, whether or not you carry a allele or not, will give you a unique risk score for Alzheimer's disease. And as you can see, the centenarians have a lower genetic risk score compared to the Alzheimer's disease patients. And you can also see the strong effect of the ApoE allele. So the different ApoE alleles have different risks, and you can see that carrying two ApoE4 alleles translate to a much higher risk compared to carrying an ApoE2 or 3 allele. But we already know that we have the highest effect size, but still there is an overlap. And we want to see if we can get that, can we really distinguish between these two extremes based on the genes. And for that, we need to fine map the GWAS loci. So we're going to generate long-read sequencing data from 300 Alzheimer's disease patients and from 300 centenarians. And we expect that the Alzheimer's disease patients will have long repeats in the regions that are important for Alzheimer's disease, whereas the centenarians will have short repeats for the regions associated with Alzheimer's disease. And for those regions, we will look into further. What we need to take into account is the age-related clonal hematopoiesis, because we will be sequencing in blood. What we know from blood during aging is that the blood stem cells will acquire more and more somatic mutations, and that the blood will become more and more clonal with age, and this is shown here. So one clone will eventually generate most blood cells in the peripheral blood, and that's what we will be sequencing. So we cannot ignore this aspect of aging when we will sequence older individuals, such as centenarians. So we think that there will be somatic mutations that we will pick up, and we need to know, was this actually part of the germline, or did this were sequences acquired during aging? So to identify this difference, we took 10 trios. So we asked the centenarians that are part of the 100+ study if they will agree to post-mortem brain donation. And a large part of the centenarians is actually willing to do that. So we have brains from the centenarians, blood from the centenarians, and we also ask their children, who are about 75 years old, if we can sample their blood. So we have selected 10 trios, for which we have then the brain, the blood, and the child, to identify sequences that are stable or unstable across aging, and that are different in the blood compared to the brain, or that are stable or unstable in the germline. So when we compare centenarian brain with the child blood. That's what we started off with our sequencing endeavor. And the persons involved in this, I'd really like to show them, is Alex, Nico, Mark, and Sanduni. And we have also open positions. So what we're now doing is we're generating long reads, and we're aligning them to see whether or not there are short reads or short expansions or the differences in expansion. And we've really been supported by the PacBio team here. We first started off with one individual, a 115-year-old woman, and we compared her genome with other genomes. And this was actually published last year. And this was done by Jasper Lindhorst. And we found that in the subtelomeric regions, there is a enrichment of VNTRs. In fact, when you focus on the VNTRs with a GC content of more than 60% and with repeat patterns longer than 15 base pairs, we see an enormous enrichment there. So that is a 21-fold increased, or actually enrichment of VNTRs in these subtelomeric regions. It is interesting that the genes that lie here are generally long, and long genes, by virtue of their length, can have more VNTRs. And long genes are more likely to be expressed in the brains. So this gives even more reason to focus on VNTRs in neurodegenerative diseases, such as Alzheimer's disease. So what we saw is that these GC repetitive sequence motifs in these subtelomeric regions are recombination hotspots, that they are genetically unstable, and that they have high meiotic recombination rates. So, and that there's specific gene families that lie there. So for us, all the more reason to look further into this. So we started to sequence our large data set of long read sequencing. So we have until now, so we're going to sequence 1,200 SMART cells. We now have sequenced 156 SMART cells, and we started with our pilot experiment for identifying the somatic changes. So the brain, the child, and the blood. That is what we started off with. But now we have also started to sequence the Alzheimer's disease patients. And this is Sanduni Wijsekera, and she's been doing a fantastic job on sequencing this and starting this whole project. Because it took a little bit of a time for us to get to high yields. We started here in June of 2021, and you can see that the yield here in gigabase pairs was very variable across SMART cells until we had actually a bit of a dip in the beginning of this year. And then we implemented several changes in our protocol, and this was with the help of PacBio. And you can see that from then on, the yield became higher and higher until we are now almost at 600 gigabase pairs per smart cell. So this will allow us to sequence 2 smart cells per individual, and then we will have at least 30x coverage with hi-fi reads, which is what we aim for. So when we look at the pilot experiment stats, so again, this was part of when we started to build up the cohort. So not all smart cells had a very high yield, it's very volatile, but we can see that the median read lengths of the hi-fi reads is 17 kb. So these are the different centenarians. So this is the blood, and then the brain, and then the child DNA for all of the trios. And then we can see that per smart cell, the hi-fi reads have generate a median genome coverage of 7x. And when we take all of the smart cells together, we see that we now have a hi-fi genomic coverage of 17x per genome. And again, we aim to generate about 30x hi-fi coverage per genome in the future. So we will sequence some additional, specifically for the brain, we will sequence some additional smart cells. So what do we know so far? When we look at the different genetic regions, we see that, for example, when we look at the ATN1 region, we see that there's a CAG repeat there. And we see that, that these are very different in different individuals. So there's different haplotypes, there are different repeat lengths there. And we also want to look at whether or not we see differences in the blood and the brain, but we have to look further into that. But there's other regions, for example, here, a repeat in the ATXN2 gene. This is a GCT repeat, and there are only 2 haplotypes there. So it's very different per region. We are now interested in identifying which features per repeat are associated with being, having different repeat lengths per read, and which associate with being more stable. Here you can see a TTCT motif, which is 170 nucleotides in the reference genome. And here you have a 32 nucleotide motif, which is also 170 nucleotides in the reference genome. And they look very, very different when you look at all the reads. So these are, so here you can see the blood, brain, and a child from our trios. And here you can see the genomes from the HPC consortium. And you can see that in both, actually in all of these genomes, the read length or the repeat lengths are very different per read. So apparently there is a somatic change or maybe technical errors in this specific repeat. Whereas when you look at this repeat, everything is extremely stable, and you can really see 2 haplotypes here for this region. So we want to identify which features associate with being, being more vulnerable for somatic change or error, compared to which features are associated with being stable. Basically, we see that about 72% of all repetitive regions are homozygous, and that is true both for the HPC genomes, so that which are publicly available, and also hi-fi read sequencing or PacBio sequencing. And compared to our centenarians, we see that about 19% is heterozygous, and that 19% has more than 2 haplotypes. So that may actually be mosaicism or somatic changes, but it could also be technical error. So that is where we are right now. Our future plans are that we want to identify germline repeat lengths associated with Alzheimer's disease, so that we can use these for AD risk prediction. We want to identify critical repeat length to see which length is associated with an increased risk. Then we want to identify somatic changes associated with Alzheimer's disease. These may increase with age, and they may also confer risk to Alzheimer's disease. So we want to identify the features of somatic instability and to distinguish them from technical error. So to end off, we would like to generate new insights in Alzheimer's disease, We want to generate a list of validated Alzheimer-associated repeat sequences. We want to generate novel insights in Alzheimer etiology. We want to improve risk prediction models. We want to identify candidate loci, and with repeat sequences, they may lend itself to be actionable with antisense oligos. Of course, we are going to generate a unique data set of long-read sequencing data, so 300 Alzheimer's disease patients and 300 cognitively healthy centenarians. We will use the cognitively healthy centenarians to generate a reference compendium of structural variants from the healthy genome, which can really be used as a reference for all sorts of diseases. We will probably generate new bioinformatics pipelines to interpret these repeat sequences. And we are very open to sharing these data with other research partners, because we realize that we're opening up a whole new part of the genome, and which is going to be so complex that we are very open to collaborating with other researchers. This is the group that I work in. This is actually the 100+ study group, and we have vacancies here. In fact, also for working with these data. So please go to the holstegelab.eu and go to the vacancies and have a look at the positions that we have open. And if it's fit for you, then please do apply. These are the collaborations that we have. We work with many people, and these are the acknowledgements and the support that we get. With this, I would like to end.